Hello, everybody. Welcome to the keynote session of Biodiversity Genomics 24. We're going to hear from two great speakers, Anne Muigai and David Chanier, who are going to talk about their experiences in biodiversity genomics and on the continent of Africa, and David in his work with the communities in Aotearoa. So these two talks are going to happen, and we're going to have questions in between the talks. So please put your questions for Anne into the Slido during Anne's talk, and Anne will answer questions afterwards, and same for David. And I'd also invite you all to stay for the musical performance after Anne and David's talks. And because we're not an in-person conference, and because we can't have a conference dinner and can't have some local band playing very loudly in the background while you shout at your colleagues and try and catch up on your lives. Instead, we have a musical performance from an amazing team called Voz Terra from Colombia, who are going to challenge us and entertain us with their view of the biodiversity of their home country, Colombia. So we're going to start this session with a talk from Anne Muigai. Anne Muigai is the chair of the African BioGenome Project, a very active and successful regional project that covers the whole of the continent of Africa. She's Deputy Vice Chancellor at the National Defence University in Kenya, and she's going to talk to us about the African BioGenome Project, looking back over the last 3 years.
Good evening, good morning. Greetings to all of you. My name is Professor Anne Muigai. I'm the chairperson of the African BioGenome Project, and I'm here to give you a presentation on the role of genomics in the face of food security, biodiversity loss, climate change and resilience, and African biogenome perspective. And this is the content of the slides that I'll be making today. So to begin with, I just wanted us to discuss some of the developments that have led to yielding higher agricultural advances in the last years. First of all, the invention of fertilizer. This resulted in a great jump in food production. And then in the 60s, we had irrigation coming in, also having a great impact on food crop agricultural output, both for crops and animals. And then the animal breeding and plant breeding concepts that began in the 1970s, of course, this had huge impacts on the production because animals were able to produce more, crops were also able to produce more. And then sustainable practices that came into place, including low tillage, and which had lower environmental impacts. This also led to great advances in animal and crop production. And then, of course, reduction in chemical farming in the 2000s, this has also led to safer ways of producing this food. But one of the questions we have to ask ourselves is why this has not translated onto the African continent, where we see about 257 million people still being undernourished, and these are about 20% of the continent's population. We have seen the amount of annual food imports increasing from $, maybe 15 billion in 2018 to 110, which is envisaged by 2025. And we see that by 2030, we will have approximately 670 million people still being hungry, not having enough to eat. So why haven't the agricultural impacts that have been seen around the world, why have they not been also available for Africa's benefits? So some of the factors that are contributing to the underperformance of the agricultural sector are listed there, including lack of crop rotation, possible poor soil management that has led to high erosions, poor road networks where foods get spoiled on the farms because they're not able to access markets, poor storage where produce is put into storage and it gets eaten up there. And of course, we're still facing the effects of COVID-19, including higher global prices. These two images here show that we have not been left behind when it comes to climate change and the devastating effects of climate change. This has, in the last 2 years, resulted in one of the worst crises that Africa has faced. For instance, between 2020 and 2023, we've had some of the worst droughts. Actually, we had 3 years of failed rains. And what these maps are showing is that how the desert areas are actually continuing to advance in this particular region. So probably the effect of climate change is being faced very, very strongly within the African continent. But while we are talking about droughts, the next consequence is the other climate change that we have faced, that drought-stricken areas are hit with large volumes of rain that has caused another devastating problem, that is of flooding. And this is satellite images that are shown over one area across the Ethiopia. You can see the Ethiopia-Somali border is dry, and then rains fall in April, and then they continue on to September, and now you can see that the river has burst its banks, and in November, the whole area is underwater. So this resulted in the deaths of more than 100 people through drowning. These are people who are used to being in deserts, and all of a sudden, their fields were underwater. The displacements of about 700 to 1 million people. And of course, these are already food insecure habitants being faced now by another challenge of too much water and too much rain. So what is the status of African genomics? So we are very, very much aware that Africa is richly endowed with a large diversity of plants and animals. These have provided a way of life for the millions of African people, and also have provided them with a mechanism through which they can practice their culture. Many communities have depended on these plants and animals for their survival. But when we think about the science bit of it, it does not caught up, because most of the plants and animals have not been studied at the genomic level. And at the same time, when this has happened, we are having biodiversity losses happening with the climate change that I've just shown you. This is also having a huge impact on the biodiversity of the African species. So what does Africa need to do? Africa needs to leverage on indigenous biodiversity, because these indigenous plants and animals have been on the continent for thousands of years, and therefore they have very many traits which are advantageous. These include disease resistance, drought tolerance, salt tolerance, pest resistance, stress resistance, so they're able to withstand high amounts of stresses. And therefore, we cannot afford to lose these plants and animals, because these genes are available for us all in the world for use and improvement of the particular species. So it's a race against time. We don't have the time to waste, because we're losing these plants and animals. The climate change is not kind on us, so that is also leading to high losses. And we cannot wait for others to come and do it for us. We have to do it for ourselves. Why? Because we can be able to prioritize, and the plants and animals that will have the greatest impact for us, and therefore will have more benefits for ourselves. So this is one of the aims behind the African BioGenome Project, a project that was conceptualized by Africans for Africa to conserve and document Africa's biodiversity. The goal is to coordinate all non-human genome sequencing initiatives, so that any sequencing initiative happening in the continent will provide data that can be able to be shared by all. And also to initiate, develop, and support any new non-human genome sequencing effort that's happening, with a goal of sequencing 100,000 plus species in Africa in the next 10 years. And the other aim is to deploy this data to conserve, to document, and also to inform policy, with the added advantage of building capacity in Africa. So the African BioGenome Project activities will cover all eukaryotic species, but will not cover any of the human genome species, because, human genome sequencing, because those initiatives are well advanced, and they're moving on very, very well on the African continent. So the question that begs right now is, should Africa commit $1 billion on genomics in the face of poverty. We are seeing that we are food insecure. We have diseases that are ravaging the African continent. We have a shortage of food. Shouldn't we just take this money and feed our population directly? Yes, we can, but we have to remember that this will provide a short-term solution, because these monies will be spent feeding the people, and therefore we will still need to solve the problem. And to solve that problem, I think it's better to look for a long-term solution. And the long-term solution means that we should be able to produce this food for future. And we have to leverage on the technologies that are available to us, including genomics, the power of genomics, the power of biodiversity, the power of bioinformatics, to be able to leverage on traits, on genes that are going to be able to give us better foods, faster growing animals and plants. And this is the reason why it makes sense to invest this money in providing for a long-term solution. So what do we envisage? We envisage that to do this, we cannot do this alone, and therefore we need to build collaborations and increase capacities. And this way, we will hope to directly contribute to the provision of genomic resources on the African continent. And like I've mentioned to you, that we hope that we will be able to push for the provision of policies that will enable us to be able to do this by providing the legal framework through which we can be able to govern this particular space. So the goal is to sequence 95% of the Africa BP genomes in African institutions. And we realize that this is a huge task, but we are ready and getting ready to do this. We also envisage that this will generate an estimated 203 gigabases of DNA sequences. And the aim is to analyze this on the continent. And immediately we started preparing to analyze these genomes on the continent. We realized that there was infrastructural and genomics and bioinformatic capacity gaps. We have to be able to address these gaps at the moment and immediately for us to be able to meet our goal of sequencing 100,000 genomes on the continent. So infrastructural gaps means that we have to develop the equipment, infrastructural capacities in African institutions. We also have to be able to train the African scientists in genomics and bioinformatics so that they can be able to be the ones to do this genomic analysis that will be generated. So to do this, we launched the Africa BP Open Institute for Genomics and Bioinformatics. You could say that this particular institute, which is embedded within the African BioGenome Project, is the training arm of the African BioGenome Project. So we want to equip scientists, we want to equip students, and we want to equip local institutions with the skills and the capacities that we need to drive biodiversity, genomics, and bioinformatics. We are very, very focused on improving the quantity and the quality of science within the African continent. And to do this, we have 5 priority key areas. Number 1, to develop curricula. We have to be able to ensure that the bachelor's and the curricula that is being taught in the universities is current and will be able to address these gaps in genomics and bioinformatics. We also have to ensure that the technology is moving at the same pace that is happening across the world, and that the institutions, the African institutions, are able to invest in provision of this infrastructure. Through this, we also want to ensure that this information change is happening at the grassroots, within the local universities, within the local classrooms, that we're able to have knowledge exchange happening. So it means that the scientists, the lecturers, and the heads of those particular programs are equipped with the current developments happening in this in this particular area. And to do this, we're going to leverage on the skills that are available in the industry within these countries, to be able to leverage on this, to form partnerships and collaborations so that at least what is available is what is very, very current. And then we want to drive the conversation on data ownership and sovereignty. How is this data going to be managed? Where is it going to be managed? How is it going to be stored? And this is one of the key priority areas that we have envisaged and we're working on. So the need to develop early career scientists is very clear, that we need to the young early career scientists to have these skills. And as we went out beginning the sampling of our genomes, we realized that one of the skills that we need is in taxonomy. We have a shortage of taxonomists on the continent. Of course, we need skills in genomics, bioinformatics, and in the areas of computer science. The other thing that we need to do is that once we build this capacity, we have to ensure that we are able to retain the African continent in Africa, so that they don't look for opportunities out there. We have to be able to place them within African institutions, and therefore these African institutions have to have the labs and the infrastructure that will be able to provide them with satisfactory work for them to do. So when we do this, we will be able to harness the skill for the country. We will be able to grow homegrown research projects, and we will be able to solve local problems. But more importantly, we will be able to provide the African young scientists with job satisfaction, proper rewards, and also be able to retain them within the African continent. And to do this, we need to strengthen the North-South collaborations and also build Africa-Africa partnerships. I think we've done very well to partner with institutions that are outside Africa, but what is very, very weak is the conversations happening within Africa. So we want to be able to have these conversations and also establish which institutions have this infrastructure. Through that, we can be able to strengthen their utilization and also ensure their sustainability. We also are very, very much aware of institutions that are able to do this and do it very well, do training, do research, do collaborations very well. And therefore, we want to leverage on this knowledge that is available, and we want to be able to learn also how they have done it. So I just want to quickly show you some of the milestones that we achieved in 2023. And this includes holding regional conferences around 4 regions of Africa: West Africa in Nigeria, North Africa in October, South Africa in August, and East and Central Africa in December. So the format of the conferences followed the same format throughout the 4 regions, where we have 2 days of conference-style presentations. And these are followed by 3 days hands-on practicals. So for the 3 days practicals, we take the shortlisted, a few of the shortlisted scientists to labs where they are able to interact with industry players, with lab technicians, lab scientists, in actually practicing some of the molecular biology and bioinformatics skills. And this was very, very successful. So we were able to register 3,700 participants. Majority of these participants were able to participate in the conference style, but each location was able to host 100 participants on site. Then from the 3,700, 400 of these received practical hands-on training in molecular biology, genomics and bioinformatics, and LC. The participants didn't have to pay for that. They only had to provide they had to pay for their own accommodation, they had to pay for their own transport, but all the other costs of the practical were met. 40% of the participants were female, so we're happy to see that there's a huge interest of genomics, bioinformatics, taxonomy, and these areas from the ladies. The conferences were attended by students, faculty members, scientists, and industry. And we were able to register participation from 49 African countries out of the 54 African member states. We were also able to launch fellowships. These fellowships are longer moments where young African scientists are able to spend periods of time. Right now, we have this particular fellowship ongoing, where our young African scientists were shortlisted, and they're going to spend 4 months on this particular fellowship. So the idea is that they spend 1 month in an African institution, and then they will spend another month in an overseas institution or another university outside their country. They are able to have longer hands-on training, and then they come back to Africa, and through this they will be able to drive sequencing of some of the African BioGenome Project species. So some food for thought, some experiences that we have had as we have gone along. We have realized that a lot of African scientists have been able to send their samples outside the country, and that's how, outside the continent, and that's how they have been able to drive sequencing of African genomes. But the question we have is actually, is this a long-term solution or a short-term solution? And as I quote Dr. Thangod Ebeneza, who's a co-chair, how many Africans do this but are not proud of it? How many Africans put their short-term personal gains before long-term African gain? Yet, African scientists complain about limited capacity in Africa, yet fail to lay the foundation for the future. So we realize we're going through the long and expensive route of laying the foundation on the African continent, but the short-term route of sending samples outside Africa will not help the continent. I think this is the best way to do it, to go the long, slow route to ensure that this is properly established. So we have also learned that training of African early career scientists needs to go hand in hand with mechanisms of how to retain them on the continent. Because if you train a young early career scientist with advanced genomics and bioinformatics skills, and you don't place them in an institution where they'll be able to work in this area, the next day is that they will be looking for an opportunity to leave. And so we have to also ensure that the infrastructure in Africa is able to sustain these early career scientists. We also need to make sure that they are well-paying positions with good infrastructure, and also opportunities for promotion, growth, and development. This way we will be able to drive genomics and bioinformatics in a sustainable manner. We also have realized the role that Africa BP Open Institute regional workshops is playing to bridge capacities in young African scientists, especially in this area, to develop capacity and training. But one of the most important things that we have realized is that we're influencing the curricula that is being taught at the universities. So this curricula is beginning to change, and therefore it's more current, and therefore it means that we are having better equipped and better trained African scientists. We are also able to provide access. There's a big movement and a big need to have these particular skills on the continent, but access to this technology has not been there. And through this Open Institute, we're able to provide access. And thank you very much. You can find more information about us on the African BioGenome Project, and I'm ready and willing to take questions. Thank you.
So it's very hard online because you don't hear the applause. So just imagine that there's applause from the audience, and there are questions. So I'll take the lead. Early on, you talked about autonomy in data. So the idea that digital sequence information was something that is and should be owned by the African nations who are generating these data. So what do you know of efforts to build such a data resource in Africa for Africans?
Yes, indeed, there are efforts to build the data infrastructure, which is lacking. And I'm aware that there are initiatives towards this effect. And we realize, because it's quite expensive to put up this infrastructure, there are opportunities to do actually one good African infrastructure that will solve the problem for the human genome sequencing initiatives and also for the plant. But we would like these initiatives to be hosted on African soil. Namely because if we do it outside the continent, we ensure that people don't get trained. And we ensure that universities and other public institutions don't make the investments. So one way of making sure that there's investment, that there's a capacity not only to build this infrastructure, to train African scientists to manage it, is to put a condition, like let's have it back home. It's very easy to ship things out. It's the easiest and the safest way to do it, but it's also expensive in the long run. But this, let me clarify that our vision is that this is open access. So having it on the continent doesn't mean that it will not be accessible to others out there. There would be an embargo as happens elsewhere, but it will be open access. Yeah.
Well, that really sounds like a vision for success. So Anne McCartney asks on the Slido, she was wondering, do the open institutes provide training on ethics, legal and social issues alongside the sequencing, bioinformatics and computer science training?
It hasn't started yet, but it's on our curriculum for next year. And this is because we've had a push for people to understand what does access and benefit sharing really mean. Having already sequenced some genomes and gone to sample, it's very clear on our documentation that LC is one of the things we're thinking about. And so in our curricula going forward, yes, it will be there. But LC, let me just say that certain aspects of LC has been incorporated into the curricula, especially when we talk about where these samples or where these genomes have come from. But I think we need a whole 3-day workshop on that, and we welcome any of the players to partner with us.
Yeah, there's an invitation, Anne, to work with Anne and colleagues in Africa BioGenome Project. Nate Jewett asks, says, great talk, great work, great vision. He wonders if there are any ways that we typically practice science that you had to approach differently to achieve the level of collaboration you've had so far in the work of the Africa BioGenome Project.
I think we had to spend some time to document what successes we want, what paths we want, before we started going out there, because you realize that we, as has happened everywhere, there's a traditional way of doing things. But we had to say that, well, this traditional way, or the normal, what we call the normal way, has achieved some success, but just in certain pockets. And there are huge areas of the African continent that has not had access to this. Secondly, we've also seen instruments, very expensive instruments being placed in public universities, public institutions, and then they just die away there with very little usage. So we had to change some of the traditional way of doing things by saying, if institution X makes such a huge investment, it's our commitment as Africans to make sure that that instrument pays for itself. So we found ourselves in a very a good position because we have a bird's eye view of sequencing initiatives on the continent, and we'll be able to connect people together. I think when the instruments are coming in, there's a project in mind, there's a collaborator outside Africa, but when the funding runs out, then everything stops. But if we are able to forge those Africa collaborations, then we can drive work towards one corner and ensure that the instrument keeps working and there is actually value for the investments that have been made.
Yeah. I mean, along those lines, Eric Jarvis has asked, so he agrees that having all the steps done in Africa is good to build capacity, but looking across the EBP, the more taxonomically focused projects or ecologically focused projects like the VGP, they have different components being done in different countries. So sequencing in one country, curation in people in another, annotation elsewhere, and the storage is in the cloud. Is the goal of doing everything in Africa too stringent?
Well, it's a good goal. It's a high target. We so far we've achieved some level of success, but remember we said that we're not sequencing 100%. We've said 95. So the 5% will take care of some of these. We recognize that there's some workflows that have already been developed, and for project-specific workflows, and we will have to send samples out. But we are pushing to have the majority here. And at least if we have the isolations, the sequencing, and some of the annotation happening here, then the rest of the other apps can happen elsewhere. Yeah.
So I was particularly impressed, let's just say, astonished is one word, but by the number of people who've engaged with your regional workshops and the number of people who've attended the hands-on genomics training. So how do you plan to keep those individuals who are obviously engaged and enthusiastic, how do you plan to keep them engaged and keep their careers moving over the next 2-3 years as you build the momentum for really getting the Africa BioGenome Project going?
It was quite interesting, and it also caught us by surprise, because when the first regional workshop was being arranged, we had a very ambitious goal of targeting 200 participants. And lo and behold, I think for that first workshop, we registered 1,200 participants. And so the question is, this is the need, this is the hunger. So we had to have very strong virtual links for people to be able to access the material online. And we looked for a big conference room that could host 100, 150 participants. So the conference style part of it wasn't such a problem as the hands-on training, because the labs where we were taking people could only host 10, 15. So out of this 4,700, I mean, in some places we pushed people to, okay, can you take 20? But they were really crowded in there. So that just shows you the need, and the young people who are training in this area. When we started out, we thought that there would just be a drop of, very few bioinformaticians, very few genomicists, but we realized it's actually not quite that way. We have many universities teaching this curricula, but the question is, what is happening to these young people?
Yeah, I think that's a key issue that we probably have to meet globally, not just in Africa, is building this workforce that we believe will, have very exciting, very rewarding jobs, careers in genomics.
Right.
But it's promising at the moment, but we don't have the funding to deliver it. David had a further question about data protection and IP. So are you going to ensure, or how will you ensure IP protection in addition to data protection? His point is that a lot of the data your project will generate about alternate crop plants, about alleles that protect against pests and pathogens, will be of interest to large crop breeding companies as well as African farmers.
Indeed, and just when we start entering into one phase, this whole Pandora's box of intricacies come up, and this is one of them. How do we protect IPs? What does access and benefit sharing really mean? And how will those IPs and incomes generated actually flow down to those people? So those are things that we're actually forging new ground in Africa, because it hasn't really worked. It hasn't really been done. And we welcome ideas and partnerships that will help us get the best route. So I don't have a solution for that right now, but it's something that we definitely will need to look at. Yeah.
And are there plans for assisting communities or groups or scientists to actually realize the benefit of the genomes that get sequenced? For example, one of the things we've realized in Darwin Tree of Life is we can provide a beautiful reference genome, but the reference genome really only has utility if there's a population genomics experiment also run, or there's a functional genomics experiment also run that actually allows people to take the raw sequence of the genome and turn it into something useful. So are there also plans inside Africa BioGenome Project to support that sort of activity?
Yes, and indeed, we did initially have lists of species that we're going to start sequencing, and then very quickly we realized that we'll have this sequence, these reference genomes just sitting idly because nobody's really using them. And therefore now we are looking at approaches where we will have major projects where major diversity projects going on, one aspect being the genome that Africa BP commits to produce. This way, we will ensure sustainability and sustainable use of our reference genomes. So those communities of African or non-African scientists, because we find there are groups out there that are working on Africa taxa, and partnering with them and leveraging on their project funding will actually help us mutually achieve both our goals. And that's an approach we're taking. Yeah,
very true. Okay, I think that's it for questions just now. Thank you so much, Anne, for that presentation and the vision of the African BioGenome Project. And I am absolutely sure that, you know, in the next 3 years, there's going to be some amazing, amazing data coming from the project and some amazing results there. So thank you.
Thank you. Thank you very much for the invitation, and I look forward to being in BG over the years. Thank you.
We'll see you again. So the next talk is from David Chanier, who's the science group leader at the Molecular and Digital Breeding Department of the Plant and Food Research New Zealand. He is interested in applying genomics, molecular genetics, to understand the genetic control of important characters in plants and animals, and especially in his role in New Zealand, where he's been assembling high-quality reference genomes and working with local communities on genomes of special interest to them. So we're now going to hear David's plenary presentation. And again, please put your questions in the Slido so that we can ask David afterwards. Thank you.
Hi, everyone. My name is David Chanier. I'm from the New Zealand Institute for Plant and Food Research. I would like to thank the organizer of the Biodiversity Genomics 24 meeting for inviting me to give this presentation. Today I'm going to present about some work that I've been doing in the last 4 or 5 years. This is a project I'm very proud of and very excited to talk about. Bit of history about me. You know, I've been doing genomics for quite a few years, about almost 25 years now, and I've been working a lot with species that are of interest to the primary production in agriculture. But recently, I've been doing a little bit more work in species that are a lot less around that species for human consumption type of work, but more about, you know, like native species and natural estates. So it's quite exciting for me to be in a conference like the Biodiversity Genomics Conference, because I'm quite used to talking to people who works in the agriculture sector, and I really see a real potential in looking at different problems but with very similar solutions and similar problematic between the two sectors, which tend to work, you know, in isolation. But I think there's a lot that we can learn in common. So that's my introduction about why I'm doing this in a way. My first slide is actually a bit of a disclaimer slide because the work I'm going to present about's got some obvious indigenous importance. And I would like to start my presentation by acknowledging and attributing this work to the indigenous peoples of New Zealand, and in particular one of the tribes. It's called an iwi in New Zealand, and that tribe is Rangitane o Manawatu. So that's a local tribe whose traditional lands are in the region where I do my research. And this group, I'm going to talk a lot about the importance of this indigenous group and what they've done in this research project. But first, I'd like to acknowledge that the samples, the data that I'm going to talk about is attributed to them. They consented the use of those samples and this data for the research, and they acknowledge that they're open to commercialization and collaborations with researchers and enterprises around the world. But really, if anyone is using this data set, they need to talk to them. So this is what those labels are about. All right. Second slide is also about acknowledging the people and the team that has been involved in this project. And that's been actually multiple entities involved in this research. And foremost is the local iwi, the local tribe Rangitane o Manawatu, with various people involved in the design and in doing the research. I also would like to acknowledge my PhD student, Colin Balkwill, is the one with the bright yellow high-vis jacket on that picture. He's been working on this project for the last 3.5 years, and he's been doing a really brilliant job at every aspect, from Maori engagement to the analysis. And also I've got colleagues from plant and food research who have been involved in maintaining and growing the relationships with our Maori partners, doing analysis and looking after plants. So here's the plan of my talk for today. I'm going to talk about what's problematic, like what sort of issue do we have to solve together with our Maori colleagues. I'm going to talk about the indigenous context and all the considerations that are important before doing the research, and in particular looking at a Maori perspective, so the indigenous people of New Zealand. And then I'm going to talk about what we did about building genomic solutions with this Maori group, and how, after building the knowledge, building the genomic tools and resources, how did we do about applying those tools to the problems we identify by talking to our Maori colleagues. And then I'm going to take a step back again and just talk about, about my reflections about partnering with a Maori group. All right, so here's the problem. This is how everything started. So just to locate where I'm located, and that Maori group is located, we're at the bottom of the North Island in New Zealand, in the region called Manawatu. And in April 2017, that was quite a while ago, there was a few massive slips that happen on the road that is linking the east coast and the west coast of the North Island. So there's a mountain range going across that island, and there's only a few routes you can use to go across. And one of them was a gorge road, which was actually the most the fastest and the most convenient. However, it was very fragile, and it was ongoing issues with slips when there was weather events. And in 2017, there was a massive one that the local council deemed way too big to deal with, because it was recurring and it was actually too big to fix anyway. So since then, we haven't had any fast solutions for going east to west. We've got, some roads that are quite windy and dangerous, and yeah, so that's the situation for us. And one of the solutions is that in the last few years, there's been a new highway that is being built across the ranges. It's called the Te Hau Aturanga. So it's the highway that goes across what we call the Tararua Range. And this is a picture here of the example of the building of the highway. And in the background, you can see a little bit the old gorge road with few of the slips that are along that road. Now, something quite interesting about that highway is that during the early stage of the building consent and design, it was clear that one section of the highway was going to cross a small patch of swamp and put it at risk. And in fact, that little swamp, which I circled in red on that slide, is actually a small remnant population of a tree species called swamp mare. And the local iwi, that is from Manawatu, has estimated there was about just not more than a dozen adult established trees in that little patch of swamp. And obviously, you see from this picture, this is quite an advanced stage of the building process. That swamp is just underneath one of the viaduct, and it's really at risk of losing all the wildlife that's in there, including the trees. So the trees, the trees that are located in that swamp include swamp mire. So that's a species that is part of the Myrtaceae family. So the Myrtaceae has many species, including eucalyptus and guava. And swamp mire, it belongs to the genus Syzygium, which is one of the tree genus which has the most species. There's more than 1,000 species of that genus. And some of them have some economic interest. For example, clove is a Syzygium. But interestingly, there's only one species of Syzygium that is endemic to New Zealand, and that's called Syzygium myrrhe. So that's that swamp myrrhe tree is the unique endemic species of the genus Syzygium in New Zealand. And because there's been a lot of habitat loss, swamp have been reducing across the country, this tree has been listed as nationally critical. So it's threatened from extinction because its habitat has been reducing a lot. One other issue that swamp myrrhe is facing is that there's a fungal disease caused by Ostropocina psidi that's called myrtle rust. And this species, from the test we've done on seedlings of these species of swamp myrrhe, we've observed that it is highly susceptible to that fungus. So we've got a problem in loss of habitat as well as susceptibility to the disease. Now, this species is quite important. It's quite a high, large tree that dominates the canopy when it's present, and it bears fruit that are important for the rest of the ecosystem, including the New Zealand wood pigeon, the keriru, and I've pictured one of them at the bottom here. Now, what is the extent of the problem in terms of habitat loss for this species? I'm giving here the example of the Manawatu region, so that's the lower part of the North Island in New Zealand, and some of the modeling that was done about the extent of historical wetland in the region that was done by my colleague Angel Osei, is that there was more than 1,300 square kilometer of swamps in the region. But nowadays, those swamps have been drained for various agricultural applications, in mostly dairy farming. And basically 99% of the swamp land that existed prior to European settling have actually disappeared. So we've got a very, very small amount of swamp left in the region, which means that the habitat of that tree species has reduced drastically. So swamp myrrhe absolutely need to keep wet feet all the time. You know, it cannot live if there's a period of dry soil. So it absolutely is dependent on living in a swamp. Now, so that's the context about the species that's endangered in our region and some other parts of New Zealand. Now, what about the indigenous context in our country? So New Zealand is quite particular compared to other countries in the world. Most of you may have heard of the Nagoya Protocol, which is recognizing, you know, the importance of benefit sharing with indigenous communities around the world. However, New Zealand hasn't signed that protocol because one of the founding documents of New Zealand that was signed in 1840 is actually quite strong in similar aspects of the Nagoya, not similar, but not exactly the same as the Nagoya Protocol. Basically, that treaty that was signed between the English Crown and Maori chiefs says that Maori actually have the right to apply guardianship, which is called kaitiaki tanga, and exercise sovereignty, which is tino rangatiratanga, over their treasures, which is called taonga. So I'm going to use those Maori words quite a lot over the talk. And as a matter of fact, as an endemic species, swamp myrrhe is considered a taonga, a treasure for Maori. Therefore, Maori have the right to exercise guardianship over this particular treasure. So that's from the Maori aspect. From my institution context, we had to recognize that we've got to work following the principles of the Treaty of Waitangi, as well as, you know, because my institution works internationally, we've got to make sure we're compliant with the Nagoya Protocol. So we developed a set of principles for working with native species, with treasured species like taonga, as well as traditional knowledge, which is called Matoranga Maori. So basically our principle is that any project where we involve native species with Maori, we absolutely have to have engagement with Maori community. And that engagement must be done with the rightful guardians of those species, and has to be done prior to doing any work. We also want to develop some partnerships, which means like early on in the discussions with Maori communities, we need to discuss about benefit sharing and how are we going to achieve benefit sharing before, during, and after projects. The last point of our principles for working with native species is about the fact that when we generate data and analyze data, we actually don't own this data. At best, we're custodian of those data sets, and that we do not have ownership or sovereignty of this data. This is held by the Maori guardians. So we really want to make sure that our status is clearly as trusted custodians. So some other tools that have been developed for researchers in the last few years include some guidelines for doing research with Maori on native species in New Zealand. It's called the Te Nohonga Kaitiaki Guidelines. So this is quite a useful document that goes through all the different worldviews and Maori values and how they interact with genomic research and what sort of considerations researchers should, think about before starting a project with Maori on a native species. There is also, we've developed in New Zealand a data repository to store genomic data from native species, because in many cases, our Maori communities and partners do not agree on data being deposited in database overseas, because once it's gone out of your shores, suddenly you lose control and sovereignty on this data. So a lot of the conditions often that we hear from our Maori colleagues is that the data should be held in a server, in a computing system in New Zealand, and the access and the reuse of this data should be through reconsenting. Another set of tools which I mentioned in my introduction slide are the bicultural labels and notices. Basically, this context is about adding some metadata fields that attribute data, data such as sequencing data to the communities, to the indigenous community who have consented and who have rights over those particular data sets. So there's different types of bicultural labels which the indigenous community can apply to the data set, and those tags can be put into reports, publications, various databases, and so on. And this metadata field is to make sure that the data keeps attributed to the communities in the long term. So if you want more information about that, I encourage you to look at the local context website. Right, at a local scale, so as I said, Rangitane o Manawatu is the local iwi, the local tribe that has sovereignty over a large stretch of land in the region. So within that region, that rohe, Rangitane o Manawatu engages with various processes, including natural resource consenting, across many activities, and that includes the activity supervising the construction of the highway and all the ecological offset that needs to be put in place for in relation to the building of the highway. Now, Rangitane o Manawatu has a principle called Rangitane Nuirawa, which is a principle of actually working to ensure the species of significance to their tribe is persisting through time within the ecosystem. So basically, those species are important for the well-being of the community, their community, and all the people living in the region. So that's their role. They feel they've got that responsibility to make sure that those species persist through a long stretch of time. And also Rangitane o Manawatu has the right to exercise guardianship kaitiakitanga over the species. and as well as the samples we take from the species and the data that's generated from it. Now, I'm going to show you an example of how we've been using genomics to contribute to this guardianship, to this Kaitiakitanga, and how it helped with the species restoration for that particular species. So I'm going to talk about building the genomic resources, which is for a species where we didn't have a lot of scientific knowledge prior to that. Understanding the species genetics in the region and across New Zealand, and then using that information for developing a strategy for the restoration of that species in the region. All right, so we started the process obviously with sampling. We needed sample to do our research. And yes, we're working on a swamp species. So we had to go into swamp places. And I've got to say that was a really exciting experience for me. So here again, you've got my PhD student, Colin, on the right, on the picture on the right, looking for an elusive species. So a big stretch of forest in a swamp where we know we probably have a dozen of those trees hiding somewhere. So that was quite a big task. And I've got to say that having our friends from the local tribes, knowing the land very well, was extremely useful for us to locate those trees and being able to sample them. Without them, that wouldn't have been possible. So we ended up identifying 3 different sites where we collected some trees of swamp mire. So there was the site called Eco Viaduct, which is that little site that's under the bridge, that I showed you under the highway. Then there was one site, Otangaki, which is basically a large domain close to a small town. And then on the other side of the ranges, there was another site called Te Waha o Te Kuri, which is again another reserve, a natural reserve with a small patch of swamp where a few of those trees were located. So across those 3 different sites, we identified 29 established trees. So that's more than the dozen we were expecting at the beginning, and that was good news. But that's still not a huge number. And you can imagine if one of those sites is damaged by the building process of a highway, we've got a problem. So we did a genome assembly for that species. We picked one of the trees from those 29 that we collected that day. And fortunately, this is a diploid species. So it only contains 2 sets of chromosomes. It's quite a compact genome, about 300 megabases. And at the time, we decided to use the PacBio high fidelity sequencing technologies. And with one run, we obtained about 49 times coverage of that species. And we assembled it using the Hi-Fi ASM package. And we had quite a very contiguous assembly. In fact, a very small number of contigs were covering most of the genome, and 4 of those contigs were actually telomere to telomere. And that assembly was very, very complete with a BUSCO score greater than 98.8%. So this is a depiction of the swamp mire genome. So it's got 11 base chromosomes. So the swamp mire genome is the one in brown on the left of that particular graphic. And on the right is another species of syzygium. It's clove. And basically, we've done synteny analysis to compare the chromosome of both species. And the synteny is quite high, very few small rearrangements, but it's very, very highly synthetic. And on the right, you can see my PhD student and Paul from Rangitane. They're holding the trunk of that tree that we used for the genome sequencing. So that's that particular tree we used. Now, something very exciting, and there was some media coverage a couple of weeks ago about it, is that we thought, what can we do to make sure that that genome sequence that we produce for that species stays, attributed long-term with the local tribe that supported this work? And with what we did about a year ago is that we thought maybe we should have a conversation with Rangitane Manawatu and some of the elders from that community and ask them if it would be, they would consider actually naming the genome. And they said yes, and a few months later, they came up with a name for the genome, which is Nahuha Ote Ia Whenua. So the translation is more or less, the the fruit of the land, and it speaks of the lay of the land, its waterways, the life force, its sacredness and freedom from restriction. And we've got a Maori description of that particular name. But I'm extremely excited, that in the long term, that genomic resource will keep the name that was attributed by the local community. And we believe that's the first time this has been done. So we did some analysis of the genome. Some analysis we did was about understanding some of the classes of gene that we thought would be important. One of them are the NLR class, which is actually a set of genes in plant that are involved in the great cognitions of pathogens and triggering resistance in those plants. So we've identified it's not 42, it's actually 421, I think, genes in the swan marrow genome. And this is preliminary. We hope that some of those genes will be the ones coding for resistance to myrtle rust. Just to continue on the theme of myrtle rust, we had a set of seedlings from that that we grew, and I'm going to talk about it later. And what we did is that we did characterization of the chemical composition of the foliage of those seedlings using HPLC, and we characterized more than 1,500 chemical features. And then we did genome sequencing for all those seedlings, and we had more than 950,000 SNPs detected across the genome. And we did an association analysis for all those compounds, and we found some hits. So we've got now some SNP markers linked to the composition in secondary metabolites, and some of them we hypothesize will be involved in the process of tolerance to myrtle rust. Again, this part of the work is preliminary, and there's more work to be done here. All right, so we've built a genome for the species from a tree from the Manawatu, but then we wanted to know also what's the genetic diversity of that species in its entire ecological range. So we did some sampling across New Zealand, and we sampled 269 individuals of that species. And then we generated whole genome resequencing using paired-end sequencing at about a bit more than 7x coverage for all of them. And that enabled us to identify more than 764,000 SNPs across the genome. We actually plotted the SNP density across the different chromosomes, and that's the graphic on the right. And we found that actually some regions of the genome tended to have a high density of variants, especially around centromeres and around the telomeric regions. So using this data, what we did was looking at the population structure of the species in New Zealand. And what we did was that we used PCA and DIPC analysis, and both approaches indicated there was probably 5 different ancestral populations of swan mare across New Zealand. And those different subpopulations match very well the geographical distribution. Now we did some admixture analysis to try to see, is there any mingling between those populations? And with, 5 different subpopulations, we see that there's some degree of admixture happening. For example, in the region called Bay of Plenty, that's with a purple dot called Tauranga on the east coast of the North Island that you see on the graphic on the right, tends to have some admixture with the other populations from the lower North Island. And also the region where I'm located, the Manawatu, that's in yellow on this graphic, tends to have, clustered together with the Wellington region. They're very similar in their genetic compositions, which means that in terms of if we needed to transfer genetics between those different districts, that would be fine because the genetics are quite similar. Another type of analysis that we've done is that we collected this set of 269 trees across the country. And what we can do is basically we can extract the environmental variables for those sites where we did the collection and use this as a variable for doing genotype environmental association. And what we found is that, in particular for a variable called precipitation seasonality, we found that there was a lot of SNPs that were actually showing some difference between the different provenances. 205 of them were in common between two different statistical approaches that we used to detect them. And they were in different parts of the genome. So again, this is quite preliminary. We're wondering, are we going to use this information to actually select plants for restoration, especially because that would probably be useful to use this information to predict what could be the response of the swamp mare populations to climate change, for example. But also, I'm very curious to look into the genome of swamp mare and looking at what genes are underneath those particular SNPs that we found associated with those variables. All right, so we've built some knowledge about the genetics of swamp mare. Now, how to apply it in a real situation. So remember what I said, we've got a situation where a highway has been built and we need to restore the trees, where the highway has been built, especially that little patch that's squared in green. As you can tell, between, if you look at images from 2007 and 2023, the whole vegetation around that region has changed quite a lot. So there's been, increased habitat loss. As a matter of fact, we learned last year that that little patch of swamp that's just underneath the highway, some of the engineers involved in the building process have poked a hole in the swamp and drained it accidentally. So we're starting to see some signs of trees dying because the place is way too dry now. So we thought we're going to have to do something quick and help save those trees as soon as possible before that genetics disappear. So we went back to the sampling site where we were. Between the time we did the first initial sampling and that sampling that we did last year, there was actually a few trees that had died because of the swamp being drained. As you can tell also from the picture on the right and on the left, both of them, there wasn't a bridge at that place or a big pile before. So the construction site has really changed the environment very much in this little vulnerable patch. So what we did is that we collected fruit from these trees to grow seedlings and replant those seedlings. So this is what I'm showing here. We've brought back seeds to our research facility, and we grew seedlings from them. So we had a set of 136 seedlings, which were grown in greenhouse, as you can see here. The picture on the left is quite interesting. This was, an example of including cultural aspects into the research. This is on the left, on the very left, is my lab technician who did the DNA extractions for that study, and on the right is Renata from Rangitane. And basically that day, we had to do a Maori song before we collected the samples, and recognizing that there was a goal for this particular sampling that it was, saving the species and improving the well-being of the community. But we had to respect, and apply those cultural practices in the process, the usual process that we've got for sampling for DNA extraction. So we grew the seedlings, in greenhouse. Some of them were replicated clonally. So we have multiple copies per seedling. So we did that with tissue culture, and that was quite successful. And then what we did from the 136 seedlings is that we did also some genome sequencing using the same method as I showed before. So we had about 900,000 SNPs across the genome that we used to look at the population structure of those seedlings and how do they relate to the adult trees that we collected from those sites. And we calculated also the kinship, the relationship between those seedlings, as well as the level of inbreeding that was in there. So we used the genomic information to actually select which were the best set of seedlings to replant based on maximizing their genetic diversity, making sure that we represent the full diversity of the species in the region, as well as minimizing the amount of inbreeding present in those trees, just to make sure that we keep high adaptive potential for the trees that we will replant. So we had genetic data for those seedlings that we wanted to replant for the restoration program. And we brought that data into a set of meetings with Rangitaneo Manawatu and various researchers. And basically that meeting was very instrumental in terms of discussing which plants we should replant and when and how, as well as identifying which sites in the region would be suitable for as sites to receive this plant material for replanting. So what we did was an ecological analysis of the full region to try to identify areas of the region that have high flood concentration and wetlands, and that would be the regions, those areas where we would be doing the replanting. So that was quite an exciting process involving the local indigenous community as well as a set of scientists to try to work together to try to find a practical solution for replanting and restoring this species. So the good news is that after all that process, that was in July of this year. So the little seedlings I showed you in a previous picture have grown quite a lot. You see them on the right. They're more than a meter tall. And our researchers, so my colleague who's taking care of our research greenhouse here is on the left, looking very happy because he's produced some healthy plants, and now he's passing it on to that team from Rangitaneo Manawatu who put that set of plants in their vans and went to replant them in 3 different sites that we had identified for replanting. So that happened, back in July during the Maori New Year. That's called Matariki. So it was quite symbolic also to replant those trees at that time of the year. So very exciting to go through the process of, starting from building a genome to actually replanting and restoring the habitat and saving a species from extinction. All right, so here's my concluding slide. I'm very excited because I believe this project is quite a good example of how we can use modern state-of-the-art genomics technology in a species where we didn't have any scientific knowledge or very, very little prior to the project, and how this project can be done with and for Maori. It's really exciting to say that, this project was really led and co-designed by the Maori community, by Rangitaneo Manawatu. My team was a subcontractor. Rangitaneo Manawatu secured some funding to do this research, and my team, the job was actually to, generate the data, crunch it, and co-interpret the data together with Rangitane. But, even if my team was subcontracted to do this work, there was very much research that involved the Maori community at every step, from the sampling to analysis and interpretation of the result. It's very exciting that, you know, that genomic knowledge, it's not an academic, purely academic exercise. It's really been very, very practical and applied for the restoration of that treasured species, that tanga, in the region, and that enable Rangitane or Manawatu as an iwi, as a local tribe, to apply their guardianship, their Kaitiaki tanga, for that particular tanga. What's also really important is that throughout the process of this project, Rangitane or Manawatu exercise their sovereignty on the data. For example, the digital sequence information that we produce, or even the plant identifiers and any phenotypes we generated, chemistry or GPS coordinates, etc. They have sovereignty of this data. So they've done that by applying that concept of biocultural labels. They also control the access of the data. So if you want to access the genome for Swan Mare, you can. However, that genome is stored in a protected repository, and Rangitane or Manawatu. So if you want to access it, you've got to fill a form that goes to Rangitane or Manawatu, and they will decide if they consent or not access to the and reuse of the genome. And again, what's very exciting through the process is that we've Rangitane or Manawatu agreed to name that genome, which means that it's, you know, it's Ia Fenua. The chromosomes have been named according, you know, IW meaning Ia Fenua. For it's going to stick forever. I'm quite excited by that. This is quite a strong move from that community. Now, this is an exciting project on a particular species, on Swan Mare, and that project was one of a set of projects that my institution, Plant and Food Research, had with Rangitane or Manawatu. This project started as one of the pilots, but the relationship between the Maori community and my institute has grown stronger and stronger in the last 2 or 3 years, culminating back last March into the two entities, the local Maori community and my institution, signing a treaty partnership. So a research and partnership that actually is based on the principle of the Treaty of Waitangi, which is quite unique and exciting. So that's the picture we've got here, where we've got the board of director, the senior leadership team from my institution, as well as the leaders from the local community, you know, posing for a picture in front of the marae, the local Maori traditional house. The relationship is so strong that we've got some projects ongoing on other species. One of them is on the top right. This is a species called Karaka, which has got some, it's a species that traditionally has been used by Maori as a food source. So we're working with Rangitane or Manawa to understand the genetics of that species. In fact, I was there with them, with the team from Rangitane or Manawatu in the field this morning, tagging trees and doing some observations on flowers and so on. So it's quite exciting. The other example on the bottom right is, you know, my colleagues from the chemistry lab and plant and food research, looking and helping with the repropagation of and chemical characterization of a species that is used as a traditional medicine for Maori. And again, this is a project that was established in response to a need and a request from the Maori community. So quite exciting. All right, so once again, I would like to acknowledge my colleagues from various institutions, including Rangitane or Manawatu, for their input, their crucial input in this project. I would like also to acknowledge the different types of funding that we've got. from Rangitaneo Manawatu for their time and the effort that they've put in there. Plant and food research, Victoria University of Wellington, where my PhD student Colin is studying.